So what we're going to be looking at today, what I'm doing is I'm going through and evaluating some of the material that's on some of the quizzes. And I can, as I do this, I can see that there are some topics that seem to be causing, several or many students the same problem. So what I thought I would do is we're going to go over some of the material that was from Unit 2, Quiz 3, that seemed to be causing students some problems. And this covers like about 4 or 5 questions because they kind of all are interrelated. So before I even get into that topic that's causing the problem, I just wanted to look at a little bit here and kind of do just a brief review on DNA. We probably all know where DNA is within a cell, and there's that nucleus. So they kind of pull out the nucleus there. And within the nucleus are the chromosomes, and I think probably most of us know that the chromosomes carry the genetic information that is needed for all body processes. So they've enlarged a portion here of the chromosome, and this is what it looks like. Probably looks very familiar to you. We know that it kind of looks like a ladder, with steps and the sides that has then been twisted. And I think we've already studied that the sides we know are made up of sugar and phosphate. And then we have 4 main bases that make up the steps in here. Those are the A, T, G, and C, the adenine, guanine, cytosine. But we kind of when we're talking about them, we just call them the A, T, G, and C. And I think we know that A always matches up with a T, and the G always matches up with a C. And the way I remember that is by thinking of a capital C and a capital G. Look very much the same except for that one little addition that's on the capital G. So that always reminds me that C matches up with G. And so if C goes with G, we know that A goes with T. Now, we know them as the A, T, G, and C. Of course, which sometimes we don't even think about this, is that, like, here's the T, thymine. There's the A. Remember the A and T go together? And the cytosine, the C, and the guanine, G, go together, that they actually have, the bases actually have a chemical makeup too, as you can see here. Okay, even though we don't go into great depth to even study at all at this level what the chemical makeup of those bases are. But if we look at the sides, and you can see this particular ladder is untwisted. See the P with the O. That's the phosphate group. Remember how it's phosphate, sugar. Phosphate, sugar. Same thing on the other side. Phosphate, sugar, phosphate, sugar. So the only thing I really want you to take away from here, I've noticed that quite a few students are getting confused on whether proteins contain genetic material. Well, DNA is the genetic material, and even though these look kind of confusing in here as far as the bases, we don't have to memorize those. We do know there's a phosphate, the P with the O there, and we do know that there's that circular sugar there. So if I was to look in a little bit at some chemical formula, if I was trying to determine whether DNA was in that formula, one thing I think that I would probably pick out that kind of stands out the most to me is that phosphate group. If the genetic material was in there, regardless of what this looks like to me, and it kind of might even look jumbled to you, I know that there's phosphate there. So if I don't see the phosphate group in there, I would know that, at least DNA is not part of the formula that we're looking at. So just kind of keep that in mind, because now we're going to go on to the topic that so many people have missed. So, as I said, this is Unit 2, Quiz 3. What carries the genetic code? Do proteins carry the genetic code? Do carbohydrates? Well, primarily we're going to be dealing with, they talk a lot about the proteins, and that seems to be the area that was missed quite a bit. Now, do you remember what proteins are and what they're used for? I mean, proteins are such a big part of the body processes. They can be antibodies. When you talk about antibodies fighting infections, those are proteins. When we talk about enzymes that help speed up processes, those are proteins. There are all kinds of structure that's built, you know, the body, the human body or any body is made up of. That body structure is made up of a great deal of proteins. So proteins, as you can see, are a very, very important part of everything here. So what I want to look at here, the part that people seem to miss, as I said, is what carries a genetic code, and do proteins carry that genetic code? Well, let's just take a look at a protein kind of basically here and see what it's made up of and see if we see any DNA within the protein. So DNA is made up of amino acids, and what they've done here in this pictogram is for each amino acid, and there's about 20 main ones that we're going to deal with. Each amino acid is going to be represented by a shape here. And when you put those amino acids together in the appropriate order, then you can make proteins. Now, some proteins can be very short, a short length of amino acids stuck together, and they're stuck together by bonds. They're covalent bonds called peptide bonds. We don't need to remember that right at this level. But if I have a short chain, it probably, when you get a protein, it kind of starts folding and making all these weird little folds and stuff. When you have a protein that is not doing that folding, it's usually called a polypeptide chain, but it's still a protein. So here's a real short joining together of the amino acids, making a short protein. Now, obviously, many of the proteins are very, very large, very, very big. And so again, what they're going to do is take those individual amino acids, hook them together using those peptide bonds, and making long chains of amino acids in the appropriate order to make a particular protein. Now, we haven't really looked in depth, but I'm not seeing anything quite yet that might indicate that proteins are carrying the genetic code. But just because I haven't seen it yet doesn't mean it's not true. I mean, what if the amino acids that we're joining together to make the proteins, what if they carry the genetic code? Maybe they do. So let's just go a little bit further and look. The only thing we've learned from this page is that amino acids joined together formulate a protein. So let's go over and look at the amino acids. Remember I said there were about 20 common ones that are used, and here they are, the alanine, valine, leucine. So what they give here is the chemical makeup of each of the amino acids. And remember, we'll talk about the amino acids. So here's an amino acid. There's a different amino acid. There's an amino acid. Now this one here is the same one as down here. Okay. So each of those amino acids, if I look carefully, I can actually figure out what the chemical makeup of that amino acid is. So let's take a peek. Remember when we were back here on the slide, we actually, even though we haven't done it a lot in class, this is the form that we're used to seeing it at this level. But we do know when we actually look at the chemical formula, here's the steps in here, right? There's like a T and an A joined together there. You can see the chemical makeup. Now that kind of looks kind of familiar over here. We'd have to really check to see if they're the same. But when I go back here, one thing I need to remember is that when I'm talking about the DNA or the genetic information, one thing that's part of the DNA chemical makeup here is I have that POOO. That kind of stands out to me. So let's go back and see if any of the amino acids have that POOO there. And I'm looking through and I don't even see any P phosphate there at all. So that shows us right off the bat that the amino acids that you're seeing the chemical structures for here do not include, at least the amino acids do not include the genetic information or the genetic DNA, does it? You can see, because if it did, we'd have to see that phosphate in there or that circular sugar, and I'm not seeing it. Okay, so let's see, what does this say here? DNA is a twisted ladder. Does it look like the proteins carry the genetic code? Does it? Because we're looking at individual amino acids. We know that the amino acids hooked together make up the protein. So if I'm not seeing any of that genetic information, the DNA here in the amino acids, I know that the protein itself is not carrying the genetic information. So then the question comes up, how do those amino acids know which order to line up and to be in? Well, maybe rather than the proteins carrying the genetic code, we've already seen that they don't, the DNA, which does carry the genetic code, kind of carries the information on how to make the proteins. Maybe that's what the correlation is there. So let's take a peek over here. Remember, we have our DNA is that double-stranded, and what they've done is they've split it. If you recall, when you're actually going to use the information on the DNA, it has to split. You use one strand here, and bases then come in and fit together. So this one's already, all the bases have come in and have matched up with the bases that are on the DNA strand there. Remember, this new strand that we're making, when we bring these bases in and match them up with the DNA, the new strand that we're making is called the messenger. That little M stands for messenger RNA. And so the genetic information is kind of transferred from that one strand of DNA onto the messenger RNA. So we're still taking that genetic information and we're transferring it to the messenger RNA, and that messenger RNA is actually going to take it outside of the nucleus to the ribosomes, where the information is then going to be used to make a protein. Now, what's kind of interesting is that we call every 3 bases here a codon. If you look here, one codon then gives you the information for one amino acid. Now, when we have our strand here and we're going to be making a new protein, there has to be something that kind of is the beginning and the end. So we know that anything farther this way is not going to be part of our protein, and anything further this way is not going to be part of our protein. So we have a start and a stop codon. So the AUG, when we see those bases that are in a row like that, it indicates that's the start codon. We're going to be starting to make a new protein, and then all the information after it is where the information is to actually make that protein. And then obviously, if we have a start codon, we better have a stop one so that we just don't keep going and keep adding more and more amino acids. So we actually have a stop codon. So start codon, stop codon, this in the middle here, and usually it's a lot longer. We just made it short here so that we can kind of see what the idea is here, how it works. If you take the first 3 bases, GAA, as it says down here, is the code for the amino acid called glutamic acid. Let's look back here if we see it. You see glutamic acid there? There it is right there. So those 3 bases, when they're together, they're read as being glutamic acid. Let's see, what next one, GA, how about CAU? CAU would be histidine. So remember, each codon, each 3 bases in that codon then give the information saying, bring this amino acid in. So first we'll have this one, which is the glutamic acid. The next one that'll be connected would be the GAU, which will be the histidine, and then it just keeps going until we make the appropriate protein. And proteins, remember, can have as few as somewhere around 50 bases to just thousands. Okay, so depending on how long this is in here will tell us how many amino acids are needed. So once we get all that information from the DNA onto the RNA, the messenger RNA, by the matching up of bases here, then that RNA is going to go outside of the nucleus, out to where the ribosomes are, outside the nucleus in the cytoplasm. Now, before we go on, though, did you notice one thing here? Remember, the A, T, G, and C are the 4 bases that make up the DNA. Look over here. Do you see any difference here? I see A's. G's, and you see there's a C. I don't see any T's. There's no thymine. So the only difference, you'll notice that when you have an A on the DNA and you match it up with a base, you're not going to see a T on the RNA. It's a different base. It's uracil. So it's represented by the letter U. Other than that, the messenger RNA has all the same bases that would normally match up if this DNA strand was matched with itself. Remember how it split originally? So let's go back and look at this. Proteins are large molecules made up of chains of amino acids. We've talked about that. There are about 20. Small, I think they can go down to around the 50s, 60s. They have 129 up to almost 3,000 there. The DNA stores the information for the cell to put together the amino acids in that correct order. And then we've kind of looked at that by looking at the codons. Then we can kind of tell how that messenger RNA, what the information is that the messenger RNA is taking out into the cytoplasm to the ribosomes, so that out there the ribosomes can bring in the appropriate amino acids. And it all depends on those 3 bases, and then the next 3 bases, and then the next 3 bases, okay? So we can see that, the genetic information on the DNA plays certainly a role in the making of proteins, but we didn't really see it. Once the protein is made, that protein is not carrying the genetic information itself. Now, one thing that's kind of interesting, remember we talked about the 3 bases. Here are those amino acids again, the 20 most common ones. And what's kind of interesting, remember we talked about the 3 bases that, depending on the order, will tell, when they go to the ribosomes, will give the instructions that this is a particular amino acid to be brought in. And look at this one here, alanine. If they want that amino acid alanine brought in, there are several different codons that we could use, or that the body can use, or that just the body process is going on, will indicate bring an alanine in. It could be either GCU that's on that messenger RNA, it could be a GCC, a GCA, or a GCG. All 4 of those codons tell at the appropriate time, bring in that amino acid called alanine and add that onto that growing chain. So you can see that, when you look through here, look at glycine has 4, valine has 4. So kind of interesting that there are multiple codons that code for a particular amino acid. Now, another thing I wanted to talk just briefly about too, there are some questions. Remember we were talking how proteins can be enzymes, how those enzymes are so important because they speed up the body processes without being changed themselves. They come to a process, they speed it up, and they leave, and they're still the same enzyme. Then they can be used again. Speed up that process, they leave, come back, and then they can be used again. So enzymes are very important. Now, in our reading, we did talk that enzymes are very specific. And it seemed like quite a few students missed the question on the enzymes and their specificity. Now, if you know that an enzyme is very specific, doesn't being specific means you kind of key in on 1 or 2 things. You can't fit it in for everything. That's what the word specific means. So if the question asked, if there's high specificity with an enzyme, does that mean that one enzyme can do many things? Well, that kind of contradicts the word specific. So what would you think? Can an enzyme, if the question was, if there's high specificity, which we know there is with enzymes, does that mean that one enzyme does hundreds of things? Well, no, because that's why we say that enzymes are specific. They have a high specificity. They are very specific to the job they need to do. So the body needs to have many, 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 many enzymes, probably thousands and thousands, to do all the, be involved in all the body processes that there are. So I just wanted to throw that in too. Let me know if you have any problem with this. This should help greatly if you understand what we just did. It should help greatly on a multitude of the questions for that quiz, and then you'll also see some of those questions either pretty much exactly or something related to the same topic when you actually get to your tests. So let me know if you need any help on understanding this at all, and we can actually look at it one-on-one together, okay? So thank you for coming. Bye-bye.